So the next speaker is Sajjad Garakhani from University of Tehran, Iran. System Pharmacology and Drug Discovery. Welcome. Yes, switch to the full screen, please.
Do you hear me?
Yes, but you have not full screen in presentation.
Now it's okay.
Not yet.
Now
It's okay.
It's okay. Okay. Thank you very much. I am Sajjad Garakhani, the assistant professor of bioinformatics and drug design at University of Tehran. I'm going to start my video for in this presentation, I would like to talk about systems pharmacology. As an introduction, over the past 3 decades, the predominant paradigm in drug discovery was designing selective ligands for a specific target to avoid unwanted side effects. Therefore, pharmaceutical company and scientists trying to find a selective compounds against the target. And for 50 years, the one target, one drug paradigm has been the driving force for development in pharmaceutical research. Also, this topic is the same in about disease and genes. In 50 years ago, they say that one disease, one gene. Also, this strategy allowed bringing new drugs to the market. A significant decrease in the rate of new drug candidate has been observed. The number of failures of drug candidates in advanced stage of clinical trial has increased, and the number of submissions for the FDA approval has decreased in the last decade. The reason for this attrition are essentially the results of a lack of efficacy and clinical safety or toxicity, and ignoring the selectivity of compounds against another targets. Therefore, to enrich our knowledge of the mechanism of disease and drug effect that subsequently leads to greater success for the pharmaceutical industry, alternative approaches are needed. Recent advances in chemical biology and systems biology have shown that most drugs interact with multiple targets, multiple proteins, and the pharmacological profile of a drug is not as reductionist as we believed. We know that we have some compounds with multiple targets and some drugs that can use for multiple diseases. Here you can see one example of compounds. The main effect of this drug is anti-inflammatory drug, and these drugs used as anti-cancer drugs for the second effect. Therefore, drug interact with multiple targets, which affect several pathways and networks, leading to a cascade of signals resulting in the pharmacological drug action. Although multiple activity against several targets might be beneficial, the drawback is that it can also lead to dramatic side effect and toxicity. Also, we have another example about that the second target is related to the side effect. And in this case, we say that this target is off target. Contrary to the hypothesis that a drug has only one target and one indication, one drug or a combination of different drugs can hit one or more targets, modulating multiple pathways with potential benefit for multiple disease conditions. Therefore, the strategy of polypharmacology and multi-target drug design is one of the most recent fields that scientists try to focus on for considering the chemical space of interaction between chemical compounds and drugs with all of the possible targets. Multi-target polypharmacology exploration increase with readily available information linking the relationship between the chemical and target space. For example, you know that we have a lot of databases with a lot of information. For example, Campbell, PubChem, BindingDB, DrugBank, and another, for example, Zinc database. And this databases has a lot of information and a lot of information available for scientists. One of the most important databases is a Protein Data Bank or PDB. In this data bank, protein structure is available, and there is a lot of information about complex of protein and ligands that give me a lot of information, useful information. With the increased facility for academics to get access to this data, chemogenomics approach, proteochrometrics approach, and polypharmacology approach have started to be developed. Such methods help to mine this vast amount of protein ligands interaction and allow predicting a single ligands against a set of heterogeneous targets. And by using the information of a lot of chemical compounds, it is possible to generate one model by considering the chemical space of a lot of chemical compounds and the features of a lot of proteins. In this way, centralized systems that facilitate the integration and standardization of diverse federated resources are in development. So drug action can be explored across multiple scale of complexity, from molecular and cellular to tissue and organism level over time and dose. For example, it has become apparent that many common diseases, for example, nervous systems, cancer systems, are much more complex than initially anticipated, and as they are caused by multiple molecular and cellular dysfunctions rather than being the results of a single defect. As a result, a network-centric therapeutic approach that considers entire pathway rather than single protein must be investigated. Therefore, the classic view of drug action is being replaced by a new systems pharmacology paradigm where a drug interacts with multiple targets, leading to both therapeutic and adverse effect. Systems pharmacology promotes a new way to interpret biological data. It encourages research at the systems biology and pharmacology interface. Therefore, systems pharmacology, a new paradigm that aims to understand how drugs modulate cellular networks in space and time in order to predict drug targets and their role in human pathophysiology. One of the main interests for the pharmaceutical industry is the capacity of systems pharmacology to predict in silico side effect caused by the promiscuous behavior of drugs and their targets in order to identify early drug side effect and hence reduce the cost and time of drug development. Systems pharmacology encourages a system-level understanding of the therapeutic and toxic effect of drug to determine their mechanisms of action in preclinical animal models and patients. This requires the development of novel approaches which will be able to scale the mechanistic observation on molecules to cell, tissue, organs, organism, and patient. The successful of systems pharmacology will require not only technological advances, but also cultural changes. It depends on a greater sharing, cooperation, communication, and aligned incentive. Systems pharmacology promotes that this can be afforded by the development of quantitative experiment and mathematical analysis of biological network, which can help to understand the multiple factors that influence at the same time on drug effects. Therefore, 10 years ago, the drug design has shifted to take into account the biological network in which they interact. In this way, the biological network is important, and this is the integral part of systems biology. In the systems biology, the main goal is that understanding of biological systems and analyze not individual individual components, but their interactions as well and emergent behavior, and learn new biology from the topology of such interaction network. Why the study of network is important? It is increasingly recognized that complex systems cannot be described in a reductionist view. Therefore, the actual output may not be predictable by looking at only individual components. And the whole is greater than the sum of its parts. And by studying the whole system, we can obtain more information. Understanding the behavior of such system starts with understanding the topology of the corresponding network. And topological information is fundamental in constructing realistic models for the function of the model, and create models of network that can help us to understand the meaning of these properties and find statistical properties that characterize the structure and behavior of network system, and predict what the behavior of network systems will be. The basic notation of network is that the network is a set of nodes connected via edge. Here you can see a network, a graph that the network is the fundamental base of the network is a graph. Now we know that we have a lot of graph, bipartite graph and networks. And we have a lot of networks, for example, in social media, in airline routes, in biological networks, such as metabolic networks, protein interaction networks. Another strategy is chemogenomics, started more than a decade ago with the idea to use the freshly published human genome to facilitate the identification of novel drugs for the newly discovered genomic test. The underlying idea are intellectually stimulating as well as particularly relevant, and the ultimate goal of chemogenomics is equally attractive and challenging: the exploration of all possible small molecule target interaction. In this way, it is possible to use the chemical features extracted from the ligand, and the features can be extracted from protein to repurposing of known drugs, predicting of target and pathway for hits for phenotypic screening, analyzing and designing polypharmacology, predicting of adverse drug effect, and screening biological relevant compound selection. By using a structural bioinformatics, it is possible to join with the, it is possible to use this information in combination with chemogenomics for drug repurposing or drug repositioning. In this slide, we can see that some example of the case of drug repositioning. Since I don't have enough time, therefore I ignored some of slides. Therefore, now we know that systems pharmacology is very important in this area. And one aspect is proteochrometrics. Proteochrometrics is against traditional quantitative structure activity relationship. By using proteochrometrics, we can develop one model for prediction the interaction space between some ligands and some targets by using one model. In one of my study in my laboratory, we developed a proteochrometrics model for predicting the activity of ligands against different isoforms of carbonic anhydrase. In this slide, you can see that predicted activity against the experimental. And we obtained the root mean square error for different isoforms. The results of this study published in SAR and QSAR in environmental research. And the results of this model, you can see here that we modeled by MLR and support vector machine. In another study, we used a metabolic network for extracting the most important or essential genes by studying
Unfortunately, you don't have time. We must switch to...
Finished. Only.
Please.
By analyzing the metabolic network, we identified the essential genes from the metabolic network of E. coli. And by using a structure-based virtual screening methods, we identified new compounds about the zinc database, and we use this strategy for the FDA approved compounds, and we obtained some compounds effective for targeting the essential genes in the E. coli. Thank you very much.
Okay, thank you so much.